Welcome to Neuro-Oncology, the podcast, where we bring you cutting-edge science, inspiring career journeys, and real-life stories within the world of brain tumor research and care.
Hello, everyone, and welcome to this special segment at the Society for Neuro-Oncology 30th Annual Meeting. I'm joined today by Dr. Robert Jenkins from the Mayo Clinic, a pioneer in laboratory genetics and genomics whose work has really shaped modern neuro-oncology, clinical care, and research. And Dr. Jenkins is being honored at this meeting with the SNO Lifetime Achievement Award, recognizing decades of groundbreaking work, mentorship, and leadership in the field. So today we're going to be talking about his career journey, the impact of the research, and also what it means to have achieved this Lifetime Achievement Award. So welcome, Bob.
Thank you.
So your work in laboratory genetics and genomics has had a major impact in neuro-oncology. What originally drew you into this area, and what kept you engaged over so many years?
Before I answer that question, I just want to make a thank you to all the patients who have given and donated specimens and consented for me, our lab, to use those specimens, because none of what we would have been able to do could not have been done without those specimens and the patients themselves. And you'll see tomorrow, many people worked with me, and they get almost all the credit. But answering your question, so what drew me into neuro-oncology? I finished my medical school in 1983 and went into a clinical pathology residency. Did 3 years. And at that point, I thought I was going to become a hematopathologist and do developmental hematology research, because my PhD had been in developmental genetics. And my postdoc had been in transferring stem cell cultures to the hematology lab at the Mayo Clinic. So that's what I thought I was going to be doing. A bit of a personal thing which relates to this is that I'm a swimmer, and I still swim 3 times a week. I did that in Rochester. And one day, about halfway through the hempath training, I was walking back from the Y, and across the street was the director of the clinical genetics laboratory. And he came over and talked to me, and we just walked back to the clinic. It was about a 2-block walk. What he told me later was that he had a glimmer that I could be the next second cytogeneticist at the Mayo Clinic. So I stopped hempath, did 6 months of cytogenetics, and joined the Mayo staff in 1987. Still thinking I was going to be doing developmental hematopoiesis, studying 13q- and myelofibrosis and that sort of thing. Well, in 1989, I got a call, or a secretary called me and said, "Could Dr. David Kimmel come and talk with me?" And I said, sure. So David came by. David had just finished a 2-year sabbatical with Bill and Joan Shapiro at MSKI. They were the premier neuro-oncology researchers of those days, and including like Daryl Bigner, but they were up there. So he came back, and he wanted to have sort of a research endeavor. And so I said, sure. And we collected fresh 50 tumors, and cultured them for cytogenetics, and published our first paper. That was one of the first times +7 and -10 had ever been found. And right after that, the NCI offered an RFP for collaborative grants to study the genetics of gliomas. And we applied and got the grant. And it was a cooperative agreement between Mayo, UCSF, Ohio State, and Johns Hopkins. Oh. So I got to work with Peter Berger from Johns Hopkins, Mark Israel, who was a big neuro-oncologist at UCSF, Alan Yates, a pathologist at Ohio State, and then our group, which was me, David Kimmel, Bernd Scheitauer, neuropathologist, 2 clinicians, Jan Buckner and Mark Shaw. So that was the beginning. I had done no neuropathology. I knew nothing, but I learned. And it really was dependent upon the fact that Bernd Scheitauer was one of the few pathologists who seemed to recognize the entity called oligodendroglioma. Another person was Peter Berger. And so they really influenced my thinking on that. We'll come back to what that means in, I think, one of your other questions.
Okay. So it was a bit of an accident. It was
a complete accident. It was serendipitous that I met the person who offered me my job at the Mayo Clinic. And it was complete serendipity that David Kimmel came to me. And
So the lesson is we should all swim a little bit more.
maybe. No, I think I'm going to have a couple of more little serendipity things. I think the lesson is to pay attention to the world around you, because you never actually know who you're going to meet.
Yeah, look up from your phones and
Look up, especially now, look up from your phones.
All right, look up from your phones. Okay.
Right.
So almost 20 years ago, you published the findings that this unbalanced translocation of chromosomes 1p and 19q mediate the loss of 1p and 19q in oligo. And thinking about how the molecular landscape in glioma has changed over the past 20 years, how did this discovery inform kind of the world we're in now, the current approach to molecular diagnostics in neuro-oncology?
Well, it was kind of the beginning of it. I think MGMT would have been about the same time. Mm-hmm. My lab discovered 1p deletion, 1q deletion using southern blotting and other old molecular techniques. Again, using oligos identified by Bernd Scheitauer. Another group in Germany found the 1p deletions. There was a long kind of controversy for about 5 or 6 years about which was more important in oligos, the 1p deletion or the 19q deletion. We laid that to rest, really, with the discovery of the translocation, which was again a bit of serendipity. I mean, one of my common things to tell my people in the lab was to play around in the lab, do things that I don't know about. And one of the techs knew that I had done developmental hematopoiesis, so he applied stem cell techniques to the culture of oligodendroglioma. We had cultured oligodendrogliomas with David Kimmel, but all we got was normal metaphases. What grew back was the normal reactive cells that are present in the tumor. But when you use stem cell media, those oligodendroglial stem cells can undergo 1 or 2 slow divisions and recovered the translocation. Now, we could have potentially, and then in the paper we published, we've described the fact that the people with the translocation had a really good prognosis.
Right.
And then RHAG 9402, we did the FISH on those tumors to look for 1p and 19q deletion, and that kind of cemented it by using the link to the clinical trial. We used FISH because it was just the technology of the time. To do stem cell culture on oligos as a clinical test, we would have had a lot of failures.
Yeah.
Right? So there would have been a lot of people misdiagnosed or underdiagnosed.
Right.
So we used FISH. That was the technique for many years. Then array came out, and my lab moved to arrays very quickly. We built a clinical array test. That test was 0 in 2015, and since my retirement, it's 5,000 a year.
Wow.
Of arrays, just looking for copy number alterations in brain tumors, not just co-deletion, but CDKN2A homozygous deletion, EGFR amplification, all the copy number alterations that we find in brain tumors. Where it's going, I'm convinced that NGS, maybe long-read NGS, it has the potential to find everything. Translocations, fusions, copy number alterations, mutations, and methylation features. The German group, German methylation group has shown they can pretty much recapitulate their classifier using not the same CPGs, but other CPGs that you can get when you do long-read sequencing. I think a lot of people are thinking about doing that. There are probably a few posters here about that. I have a paper that I might be reviewing, right? It's going to be something that's going to happen. I'm almost, because it's the best way to go.
So we can simplify the diagnosis a bit more because now we have so many different tests. So we have maybe a fusion test, we have the MGMT methylation. Sometimes you do the 1P19Q ahead of the NGS through FISH, for example.
Right.
And this is like a one-stop shop.
This would be a one-stop shop. And in fact, the way one of the long-read sequencing platforms works, you can actually define the regions you want to actually recover sequence from. So you don't recover the whole genome. You would recover part of 1P, part of 19Q, EGFR, MGMT, CDKN2A and B, all the mutation areas. You might only need to actually sequence 5% of the genome. That is very fast in some of these long-read sequencers and could even be potentially done while the patient is in the OR.
Right.
And I know groups are thinking about that.
Yeah.
Whether it really will be that fast, I don't know, because the technology requires the read go into the chemistry that does the reading, and then eject it if it doesn't have the sequence that you're trying to find.
I see.
So it's still going to have to sequence a lot of pieces to maybe get that 10% of the genome.
Right. But, theoretically, the speed will get a lot faster over time.
Right.
Yeah. Okay. So since this initial discovery of 1P19Q, you've published so many things, whether it's the molecular implications of the IDH mutation, the genetic underpinnings even of familial glioma, which we constantly are talking about in the field, and even assisted with neuropathological diagnosis of glioma in Nigeria. So when you reflect on your career from training through your current role at Mayo Clinic, what do you think about in terms of the particular skill set that you think was required for some of this success?
I knew you were going to ask this question, so I've been thinking about it a lot. Actually, I believe it is really being willing to think deeply and try new things. My PhD mentor left me with a couple of adages. One was pick a biologic problem that's important and hopefully tractable, and you follow that problem wherever it takes you. Don't be stuck on a methodology like cytogenetics.
Right.
Do what you need to do to answer that biologic problem, and then move the needle. And the answer you get back might send you down the path you thought you might be going, but it might also send you down a parallel or different path because it's going to maybe be it might be simpler to do, or results will come back more quickly. But you're gonna follow this path, but it's going to branch. And I basically have done that all along. We knew that 1P19 codeletion was involved in the development of oligos, but you need a TERT mutation and an IDH mutation to get the tumor. We know you need IDH, P53, and ATRX to get an IDH mutant astrocytoma. The problem that we talked about before we started was a great future project would be how does the cell know which way it's going? They're going down different paths. In more recent years, we've kind of gone down the path of astrocytoma because the modeling of astrocytoma is simpler in the mouse than for oligodendroglioma. So besides just thinking deeply, finding good collaborators and having people willing to work hard and do experiments that are going to potentially be productive.
So be wedded more towards the question and not so much to the tool.
Right.
Yeah.
Exactly. And you might have to learn a new method, or your tech or postdoc might have to learn a new method. If they can't, or your institution doesn't have the resources to do that, they can go off and to another place and learn it, or we can develop a collaborator. The finding of the germline SNP that is almost certainly the causative SNP for the development of IDH mutant gliomas was the causative nature we've more or less determined through mouse modeling by Daniel Schrammick at the University of Toronto.
So as a physician scientist, do you gravitate more towards the research side or the clinical arena?
Oh boy, I tried to keep them balanced. I think for most of my career, they were probably pretty balanced. If anything, the research side took more time because everything was changing. But with the advent of the arrays in our clinical cytogenetics, or clinical lab genetics division, and since I was the one who had invented the test, I ended up doing a lot of interpretations, a lot of time on interpretation of those arrays, 4 to 6 to 8 hours a day. So in the last few years, I was doing way more clinical work than research work.
Yeah.
Just based on the volume of the clinical work.
And did you find that equally fulfilling as the research side, or you missed doing more research?
Oh boy, I knew we were making a difference with the results we were finding with those clinical tests.
Yeah. Okay. So the fulfillment comes from the sort of the impact on the patient side.
That's exactly right.
Yeah.
The fulfillment in research comes at a much slower pace.
Yeah.
And true fulfillment, like defining the biology of how one SNP on 8q near MYC can accelerate the development of IDH mutant gliomas, that took, 4 years of animal modeling, 2 more years of all the downstream molecular biology experiments, ChIP-seq, everything else, to pull that all together to get one paper.
Yeah.
Right?
Yeah.
Well, and of course there are going to be more,
Right.
right? But they're all going to be, they're hard,
Yeah. Yeah. The timeline is much more delayed.
especially if, we all know, especially if we want to publish in a high-impact journal,
Right.
it can be a year, 2 years to get that paper through the review process. But they all benefit from that.
Right. Yeah, of course. And eventually the product is more
Really
polished.
good.
Yeah.
That's right. Polished and powerful.
Yeah.
And meaningful for everybody.
Right, right. But you have to be dedicated, right? And you have to be committed to the work for sure. So
Exactly.
So receiving the Snow Lifetime Achievement Award is a tremendous honor. Is there a particular part of your career that you're most proud of?
Yeah, I think, yes, I am. I can say that we had a long-term collaboration over the years with Margaret Wrench at UCSF and her colleague John Winke. A serendipitous thing that happened there is one day I was walking with my wife and our dog through part of Rochester, and I saw this woman walking in front of me who I recognized, who I knew lived in San Francisco, and it was Margaret in Rochester to visit a friend.
Oh.
So I knew she was doing GWAS studies of gliomas. I knew we were doing GWAS studies of gliomas. I knew that we could collaborate and validate each other's findings. So she came back, gave a talk, we formed a collaboration. We made many trips to UCSF. My lab did, and they made trips to Rochester. That interaction with building true friendships has meant everything to me. In fact, the lead statistician in Margaret's group, Annette Molinaro,
Oh, yeah.
and the lead statistician in Rochester, Jeanette Echo Paso, are best friends.
Oh.
And they got to know each other through our collaboration together. Those are the things that really mean something. And on top of that, we made good science too, right?
Right.
So it's hard to have, it's one of the joys of my life to be able to do good science and build friendships, lasting friendships.
Yeah, and sometimes those last longer than the science does.
Oh, you betcha.
So mentorship and legacy are part of what this award recognizes. So are there particular individuals that you cite as the mentors that really influenced your career journey?
Oh, I had many, right? I've been around for a while. But my PhD advisor, I already mentioned him, Albert Dorfman, he had one other adage. He said, well, you're going to be working in an area that might have 10 more people in the world working on it, and they all have your idea. Ideas in science are actually more common than you think they are. And he said, what's going to set you apart from your competitors is that you get that paper out first. He was right.
Yeah.
And we generally, I don't think it really matters if you're first and second. I think he was he was really old school, right? But he's right. The more important part of that is that there's always somebody thinking about the same things you're thinking about.
Right. Okay, so that's good advice. So.
And then the mentor that helped me do developmental hematopoiesis was Ken Mann at Mayo. Gordy DeWald was the cytogeneticist who hired me. We couldn't have done any of what we did without Bernd Scheitauer and then Caterina Giannini. Because the recognition of oligo, especially 30 years ago, was not a simple thing. And there were institutions where oligo was almost never diagnosed, and there were institutions where it was every diagnosis. Well, not really, but a lot.
Yeah.
And of course, the reality is somewhere in the middle. Bernd recognized oligo. We did the right genetic tests on those tumors that he called oligo, and lo and behold, 1p19q came about. So they're important. Margaret, I've mentioned. Two other great epidemiologists are Melissa Bondi and Beatrice Moline, who led something called GlioGene, Glioma Familial Network, and the Glioma International Case Control Study, GICC. That was a large group of epidemiologists. I was sort of a pseudo-epidemiologist, met every year for about 8 years, moving that field forward. Don Coffey, he passed a few years ago. He was one of the leaders of prostate cancer research at Johns Hopkins. At one point early in my career, I did a lot of prostate cancer research. And Don always had something positive to say to me, always. And I was from the Mayo Clinic. And we'd see each other at meetings. He got to know me. I mean, just that friendship and those thank yous and congratulations and all, meant the world to me, right? I think we all need to remember as mentors to do that more, right? And then lately, Dan Schrammick, Jeanette Ecopasso, and Dan LaChance are my collaborators. Those are the groups of people that have really mentored me almost continuously from finishing residency and postdoc to now, 37 years later.
Yeah, mentorship doesn't end.
No, mentorship does not end.
Right. So if you were advising a young clinician scientist going into the field today, what one piece of advice would you give them to inspire them or motivate them to succeed in this very difficult field that we're in?
Yeah, find a really good tractable scientific problem. It would be best if that problem also had some clinical translational relevance, but that's not necessarily needed. But find a problem that you can accomplish and stay focused on it. And be willing to shift as the data changes. Seek advice from mentors, not just your primary mentor. And finding a good primary mentor is always a good thing. I don't know really how you do that, but you have to do that.
Yeah.
And then think deeply about what's going on, not just in your research, but what's going on in the other people that are in your lab. So I have just one final example of serendipity. I was in my PhD thesis, and things weren't going very well on the project I had selected. And there was a technologist who was doing some interesting experiments, and he described to me his results one day. And I went home, and literally sleeping on it, I came up with the reason why, the mechanism of his results. And the lab I was in had the resources to do the experiments that I had in mind. And basically within 1 week, I had my thesis.
Wow.
I mean, I had to do a lot of additional stuff,
Right.
it was completely different than what I had been working on. So the problem was, the first problem wasn't really tractable, and it actually was probably wrong. And so I'd spent a year and a half on that. And the message is, lab meetings are important. And sometimes people can sit there and be very bored. But if you're paying attention, you can potentially help the person in your lab be successful, and you might be successful too.
Part of looking up.
Part of looking up, not staying out of your cell phone,
Yeah.
which is probably pretty common in lab meetings nowadays.
Yes, exactly. Or when it's your laptop and analyzing data.
Yeah, and with many of them being on Zoom, you can do a lot in the background with your eyes on the screen and not listening really carefully.
Yeah. So we all need to listen a little bit more. All right. Well, Dr. Jenkins, thank you so much for joining us today and sharing your experience, your advice on mentorship, and kind of more about what this Lifetime Achievement Award really means. And, clearly you've given so much to the field, but also have paid it forward to so many other trainees over the years. And I think the field is really grateful for the work that you've done. So I hope everyone has enjoyed listening to this interview, and hopefully everyone can enjoy this meeting and take some of the molecular genetics, molecular diagnostic education and advice with them as we go through this exciting meeting.
Yes, thank you, Evan, for having me on your show.
Great.
We hope you have enjoyed this episode of Neuro-Oncology, the podcast. The views and opinions of the participants in this podcast do not necessarily reflect those of the Neuro-Oncology family of journals or their affiliated societies. And finally, until next time, thanks for listening.